So welcome, folks. It is my pleasure to introduce Dr. Katie Fisch. She is an assistant professor in the UCSD Department of OBGYN, her obs obstetrics, gynecology, and reproductive sciences. A lot of big words. She's also the faculty director of the Center for Computational Biology and Bioinformatics, and she's the co-director of the ACTRI Center for Excellence in Translational Immunogenomics. So Katie and I have actually been working together for quite a few years now. Everyone lived through the COVID pandemic, and I'm assuming you lived in San Diego County during that. So Katie and I worked really hard to build the computational framework through which all the COVID sequences that were collected throughout San Diego County got analyzed so that we could kind of monitor the lineage of prevalence of whatever COVID lineages were kind of spreading. So Professor Fisch, she does a lot of work just generally in computational biology. So this project is not directly related to that, but it's going to be about some of the more recent work that her lab is doing. So let's give Professor Fisch a hand, and I give you the floor. Thank you so much for joining us.
All right. Thank you so much for the nice introduction. And hi, everybody. I'm Katie. So I consider myself a computational biologist. And so what that means is taking, computer science, statistics, and marrying it with biology. And that can take various flavors. So as you heard from Dr. Moshiri, we've got, COVID sequencing lineages and big computation, all the way down to image analysis and molecular analysis of various tissues and diseases. So I want to give you a little introduction about where I came from and how I got to where I am, because it was not a direct path. So I grew up in Downey, California, which is in South Los Angeles. I was pre-med, or I wanted to be a pre-med. I was like, I'm going to go to medical school. So got into Berkeley, which was great. But there I fell in love with research, and the first research opportunity I had was actually on data from hummingbirds in the Andes in South America, working with a PhD student at the time, looking to see, what the different colorations and the different evolutionary traits that they had, how those mapped to where they lived. So the various niches that they lived in, if they lived really high altitude or, lower altitudes, how that changed what they looked like. So once I got that taste of research, I was like, oh, this is so great. I'm not pre-med anymore. I'm going to go do research. I'm going to get a PhD, which was really great. Because I hadn't even considered that. That was not something I had even known, kind of really. I mean, I knew what a PhD was, but it wasn't something that I knew you could get a job doing. So that was great. And so then towards the end of my undergrad, I studied abroad in Costa Rica, where I studied mantled howler monkeys. And what that actually meant is I went around the rainforests collecting monkey poop and then taking it into, it was actually a clinical lab to look for parasites. And the hypothesis was that monkeys that lived in more urbanized areas closer to humans, with water bodies close to human activity, would result in a higher parasite load than those that were really deep, really deep into the rainforest. So that was like my last hurrah of undergrad. And so then I took some time off, which I if you have the opportunity to do so, I recommend you do, because it's important, to get different perspectives and real life experience. And then I got into a PhD program at UC Davis in ecology. So this was kind of still what I was doing, which is not what I'm doing now. Mind you, I'm at the School of Medicine doing human biology now. But this was where I got to where I am now, saying, oh, I really like these, ecological systems. I like how all these things interact. And for my PhD, I studied fish. And yes, my last name is Fisch. And so I studied fish in my PhD, which was awesome. And so, it was fate, of course, I didn't go in wanting to study fish. I wanted to study sea otters. And they're like, no, we have money for fish. So this is what you're doing. So this is what I did. But it was really great because it was a conservation genetics project. And so what that means is that this is a delta smelt in the California, San Francisco estuary. You may have heard of it on the news. It's a very politically sensitive fish species, meaning that it is an indicator species of the health of the ecosystem of the Bay Delta. And for our drinking water and agricultural water, we pump water from the delta, to drink and to water our crops. But that also sucks in these fish and disturbs their habitat. And so this is an endangered fish species. And so what my project was looking at is population structure of this fish in the Bay Delta, to see if there are subpopulations and to see if they were creating hybrids with other closely related smelt species due to their low abundance. So again, totally different than what I do now, but a really strong basis in genetics and kind of the statistical statistical genetics, background of how these different, systems interact. So then I did 2 postdoctoral research fellowships. And so this is when, once you have your PhD, you keep training. So you go, back to you go to a different program generally and get additional training in a specific area that you want to expand into. And so this is, where you go to, continue on your journey to being an academic or an industry scientist sometimes. And so my first postdoc was at the San Diego Zoo, which is, again, pretty awesome. I worked up at the conservation or the Institute for Conservation Research up in Escondido at the safari park. And it was cool. But my project there was specifically focused on population modeling. So this is where I learned to first actually do coding. So during my PhD, I worked with giant spreadsheets and it was super inefficient. And it was like, why are we doing it like this? But I don't know how else to do it. So I'm going to sit here and I'm going to make equations in Excel. But then I realized, okay, wait, where we're going in the world is we need to do this more efficiently. And so my first postdoc was just computational modeling. I started learning Python, the programming language Python, but I was sitting at the Institute for Conservation Research with no other computational people around me. So this was kind of a self-taught. I've never taken a computer science class in my life. Self-taught, struggling through this. Cheetahs did walk by my office sometimes on leash, so that was a perk. I really enjoyed that part of the job, and taking a walk through the thing at lunch. But then I realized if I really wanted to be a computational biologist, that there was no way that me struggling by myself was going to get me to where I needed to go. And so then I did a second postdoc at the Scripps Research Institute, and this was where I finally entered into human biology. And so and what I do now, so computational biology. So that was on Fisch. I forgot that. And so at the Scripps Research Institute, I worked in a computational lab. So they were computational by training. I could turn to someone and say, I don't understand what's happening. And they're like, oh yeah, here, this is how you do this. Or here's this great resource. Here's this cheat sheet you should keep on your desk, this kind of thing. So it really taught me the value of community and community of other computational folks, and also collaboration, right? Really that you can't do your work in a silo. You have to be collaborative and it's okay to ask for help. So I really enjoyed that. And so there I worked on osteoarthritis of the knee. So we worked with collaborators who actually took human knee specimens and did analysis on them. And I was the person that did the analyses. And then I also worked on breast cancer. And this was when precision medicine was a big buzzword coming out, hey, we have new molecular techniques. We're going to do precision medicine for everybody. And so we worked with breast cancer to molecularly profile and develop tools to molecularly target breast cancer subtypes. And then I got my first real job at UCSD, and I was staff. So I decided that after this kind of animal heavy, ecology heavy path, that I decided that I wasn't necessarily ready for a career directly as a professor in academia. But the Center for Computational Biology and Bioinformatics just started in 2014, and so they were hiring people like me that had PhDs but weren't going to be faculty. And the goal of this center is to collaborate with other groups, other faculty groups and labs and grad students and postdocs and undergrads to help them, to collaborate, to help them learn how to analyze their data sets, analyze them for them, right? Collaborate on these big projects to really advance science. And so I thought this was really great. It was really great. And then I was able to advance in that role and became the executive director of the center, which meant then I wasn't necessarily doing the day-to-day analyses as much as I was, but more the actual management and finances and the more business side of it, which was crazy because I'm not a business person. So again, all of these things are a lot of learning on the job. How do we do this? Well, I'm going to have to figure it out. But I feel like that is kind of a lesson in life that I have learned, is that most things you do, you just have to figure out how to do them. It's not like, oh, I took a class and now I know. It would be nice if that was how it worked, but it doesn't. So don't be afraid of doing new things, because you'll figure it out. So that was really, really great experience. But then I realized, my ultimate goal was always to be faculty, was always to run my own research group. And the experiences I had at CCPB were great because I got exposure to all different types of diseases, from Alzheimer's to COVID to cancer, very broad, biological view of things. And so then when there was a position open in the Department of OBGYN, I applied and was was able to get a position as an assistant professor in the OBGYN. So I've been in my lab for 4.5 years now. It'll be 5 in the fall, which is crazy to me, because that means I'm old, but that's okay. And so, but now what I study pregnancy, and I study specifically diseases in pregnancy and how the placenta, which is the organ that supports the baby's growth and the interchange between the mom and the baby, how that contributes to disease, and how diseases of the placenta specifically can actually cause the mom to have diseases. And so I actually think that this, for me, for my career trajectory, it seems totally far away from monkeys and fish, because it is, but the underlying thread to all these things is that I'm understanding the DNA and the molecular mechanisms behind all of these diseases and how tissues work, both in healthy states and in disease, and then applying it. So all of these things that I've done are very much applied. Like, here's a problem, how do we solve it, and how can we apply that? Whether it's changing how we manage a fish in an estuary from a political standpoint, or from like a conservation management standpoint, to how do we identify what's going wrong in a pregnancy to potentially intervene earlier or come up with treatments for it. So before I transition into other things, does anyone have any questions about this trajectory or any other things you want to ask?
Feel free to unmute, folks. Feel free to type in the chat with no pressure. Cool.
All right. Anyway, I can't see the chat, so if there's anything in there, I'd appreciate if you can.
Yeah, I'll keep my eyes on it, and I'll keep my eyes on
Thank you.
hand raised and all that good stuff too.
Cool. Thank you. All right. So now, so generally what my work focuses on, so it's pretty broad, lots of different topics, but the ultimate science question is, how do we get from the genotype, which is what's encoded in your DNA, to your phenotype, which is the disease or the color of your hair? And so I really like this graphic showing that the genome, right, there's many steps from going to genotype to phenotype. So you start with the genome, which in this analogy is like the cookbook that grandma has, that's really old and has all these great recipes in it, but it's just the instructions. So if you take the book, it doesn't turn into cookies. It's just, here are your instructions. And some of the techniques that are some of the things that you can look at in the genome, besides just the actual sequence of DNA and what's encoded there, you can look for things that are different. So you can look for genetic variation. So whether that's just in different populations have different levels of different genes or different alleles. Or you can actually look at how the genome is structured. So how the chromosomes are structured relative to one another, and how things are regulated, which then gets into the epigenome, which is okay. So I'm going to talk very briefly later about a few things and a TAC-seq. This is accessible. Looking at accessible chromatin regions. So looking actually at parts of the genome that are open and closed. This is part of like the genome/epigenome. And here now, when we have transcription, it's the equivalent of copying your recipe from grandma's recipe book to an index card. So now you're actually transcribing it, you're copying it, which is like transcription in a cell. And when we talk about transcription, we're looking at very many different things, but generally we're looking at the level, how genes are expressed, the level at which they're expressed, if there are different forms of the genes that are actually expressed. But there is a whole thing, whole field of studies just on the transcriptome. There are a few data slides I'll show later, and in the data exploration I'll have you do, looking at these various levels of gene expression. So now that you have your index with your recipe card, so your genes have been transcribed, your recipe's been transcribed. Now, in order to make a cookie, you have to combine these ingredients. And so this is where we talk about kind of the proteome, where you have the proteins that now you're made from these transcribed genes, and now you combine them together, you mix them together, which allows you, so here are various things of proteomics you can do, but then you mix them together, and finally you get your delicious cookie batter that then you bake, and now you have cookies. And so here we're talking about the phenotype, or the endpoint is the delicious cookie. In all of the studies that we do, the endpoint is, the disease or the tissue state or, other trait that we're looking at. So I kind of like the cookie part. Okay, so now we're going to talk about the placenta. And I promise that I don't have any real pictures of the placenta. I did take them out. So yes, in our field, we have very graphic images. And so I took them out. But anyway, so yeah, so this, I'm going to talk about a project that we have ongoing in the lab. And this is a very large collaborative project with several labs at UCSD and also across the country. But our focus is on the placental tissue. And what you're seeing in this image here, this is a spatial proteomics image. So it's looking at the proteins in a slice of placental tissue that has been stained with fluorescent antibodies, the antibodies targeting individual proteins, so you can identify individual cells. And I'll show you a few more detailed pictures of this later, but I think these spatial imaging technologies are very beautiful. And as you can imagine, there's a lot of computation involved in analyzing these images, right? Because you get this image off of the machine and you have to figure out what to do with it. But it's pretty exciting. So without further ado, so the placenta, just briefly on the structure and function, just so you have some context for what we're looking at later. We have the baby here, and it's connected to the placenta via the umbilical cord. Then we have the placenta that actually attaches to the uterus of the mother and invades some of the endometrium to remodel the spiral arteries in the mom's uterus. And so that's how the baby actually plugs into the mom, actually changes things of the mom's blood vessels, which I think is crazy. And then, so you can kind of see that here, which then allows more blood to flow into this space. So it kind of pools there. And then the cells in the placenta are exposed to the blood and absorb all the nutrients and oxygen and other things that are in that space. So it's also a very large organ. So, I mean, this is a picture of a baby and a placenta, but I mean, it can be several pounds at birth. So it's very large. And it's redundant. It has these tree-like structures. Someone once equated it to like a cauliflower, which I thought was kind of gross, but also proper. Like it made sense because each of the, stalks of the cauliflower is, a branch of this placental villus structure that's exposed to the blood and exchanges nutrients. So at these interchanges, so right at the cellular level, we're looking now, this is where, within each of these villi, so these cauliflower stalks here, there's the baby's vasculature. So the baby has the blood vessels, the baby's blood vessels are here. They're called the fetal circulation. So the baby's blood is here. The mom's blood is over here. So the blood doesn't actually mix together, right? It's divided by layers of cells. But then the cells themselves, which are super important, are the ones that are facilitating this exchange of oxygen and nutrients. And so what we study is these specific cells of the placenta and when they get messed up. So when things happen to the cells, so if they're diseased, if they are exposed, say, to toxins or other things through the maternal circulation, this can cause these cells to not do their jobs properly, which then can cause all sorts of problems in fetal development and the placental development, and then lead to diseases in mom. So it's a super important system. So it's super important. So in normal pregnancy, when I was talking about the arteries, the spiral arteries being remodeled. So if this is the uterine vessels, this is what, a normal non-pregnant state would look like. Then in normal pregnancy, here we have the tip of the placental villus that I was talking about. And what happens is these cells, these are called trophoblast cells, which are the main cell type of the placenta, which are like, they're epithelial cells. So they're similar to skin cells in what they are. They have these things called extravillous trophoblasts. So they turn in, they differentiate, they turn in, so they're stem cells, they turn into these extravillous trophoblasts, which are the ones that actually migrate out of the villus into the mom's spiral arteries here, and then take the cells of the mom and break them down and move them around to make the vessels bigger. So they actually do this, which is crazy because this is baby. So this is baby DNA in these cells. This is not mom's. The placenta is all fetal DNA. So it develops with the baby from the baby cells. So I find this to be fascinating. So in a normal pregnancy, this happens. You have these large arteries. Now the baby can get all the nutrients it needs through the placenta. But in abnormal pregnancies, so in diseases some you may have heard of, such as preeclampsia, is a common pregnancy disorder where the mom has high blood pressure later in pregnancy and then has to, in severe cases, has to deliver early, which of course can have issues for the baby delivering early, but if left untreated, that can actually cause seizures and death. And so it's a big problem. And what they find in preeclampsia, for example, is that these extravillous trophoblasts don't do their job properly for whatever reason. So that's what we're studying. We don't know exactly why this is happening, but they don't do what they're supposed to do. And so now the blood flow is restricted in these spiral arteries like it was kind of in normal. So the baby doesn't get the blood that it needs, but it can also cause high blood pressure in the mom, right? Because there's this pull. Imagine this pull of your blood, of the mom's blood into the placenta, but there's like little small channels to get through. Right? It causes high blood pressure, which then can cause bigger organ problems. So when we're looking at this is what we're focusing on. And so this project, as I mentioned, so this is a large consortium that's funded by the National Institutes of Health, called the Human Biomolecular Atlas Program. You can go to this website here and see all about it. They have a very detailed website, and we're going to go explore some of the website in just a bit here. And what the goal of this consortium is to profile healthy organs. So that right now, I mean, this is a slightly outdated. So we have placenta here, but we're also looking at uterine muscle and fallopian tube as well. But we're talking about placenta here. But the goal is to profile healthy human tissues and create a reference map. So scientists or high school students or anybody can go and look at cell types and what genes are expressed and what proteins, where they are in these various tissues to use as a reference, right? Because how do you know if something is happening bad in disease if you don't know what the healthy state is? And so this is a very large consortium it's actually international. So here's just showing a map of the US where all the sites are, right? We're down here in one of these, but there are several other groups in California that are part of this as well. And each group has its own focus of either technology or tissue type that they're focusing on. So of course we're the pregnant female reproductive system. But this was a picture of one of the conferences where you see all these people coming together, collaborating for this one common project, which is really great. And the goal here, besides just to create these maps, is to actually do them at different spatial resolutions and in 3 dimensions. And so for all of the tissues, we are doing single cell assays. So at various scales, so some genomics assays, epigenomics assays, looking at the RNA, the proteins, and some groups are even looking at metabolites at the single cell level. And then, as you saw a picture of earlier, we're also doing these spatial profiling assays, right, at the protein level, the RNA level, and some groups are doing it also at the metabolite and lipid level. But then what's, I think, extra unique about this consortium is that there's a large focus on the orientation and the location of where the samples were taken. So a lot of the studies that we do, because it's really difficult to do this, are just taken, okay, I have a giant placenta, I'm going to take a chunk out and I'm going to go do my experiments with it. But in this consortium, they're very specific about where, what part of the organ was it taken from, what structures were sampled, which way was up, which way was down. And so they call that, identifying landmarks, and then an orientation, because then we're looking for, specific, functional tissue units, so like anatomical structures within each of these areas, and linking that with the histology, what cell types are there, and all of these various spatial assays, right? Because in this, for example, this kidney, this part here may have different structures and different functions and cell types than this part over here. So you can't just say, oh, the kidney is the same all throughout, because it's not. And so that's what this is doing, is trying to put this in. So you can imagine at each of these levels, the amount of computational involvement that is needed to analyze each of these data types, bring them together. Image analysis is a whole different field of computational analysis, and then linking it back to the molecular states. And then this part, again, I don't do this part at all, is, this 3D modeling, and again, I guess it's some sort of image analysis, just something Nima might know better than I do, but something that they do to bring this all together to to let you see what everything is doing and where it's happening. And they call this reference coordinates, right? So they're actually putting this into a what they call a common coordinate framework, so you can very specifically find where you're looking at. And so the goal of this is to put all these data publicly available on the internet for anybody to see and to analyze. And you can even download them if you have computational skills and do analysis on your own computer for some of them, or if you have access to compute resources, like all the data that they have. So it's very exciting. And so here is an example of one of the common coordinate framework where they have the whole human body. You can toggle on and off these various organs and specific parts of organs. And then you can see within here where they have these blocks, that's where the tissue blocks that are being analyzed, that you have access to the data, were taken. So you can say, okay, I want this little corner, this little piece right in the middle, and then I'm going to zoom in and I'm going to see what's there. And so right now they have a 3D reference object library. It's available here. And when you go in, you can see here, this is a video of the kidney. Is that the kidney? Yeah. And so the kidney is the most well-developed. It was one of the first organs that they did. So it's, of all the stuff that we have, the kidney is the most well done. The placenta is still not fully on the website yet, right? Because we're still generating data. We're still getting it up. But so kidney is a good one to start with. But you can see, right? So here you zoom in, here are the different structures. Here's that yellow part is the block where they took the sample from. So pretty cool. And what's behind all of these things, and this is not a pretty picture, but I think it's an important thing to understand from a computational perspective, is that while we may have really advanced tools and AI and machine learning to train models and understand things, the underlying data that we're using really will impact what we have. So what this is all based on is something they call, it's this really terrible thing, ASCT+B, anatomical structures, cell types, and biomarkers, which is this very painstakingly manually curated by biology experts to map various parts of an organ to the cell types they expect to see in that part of the organ. And then what marker genes or proteins they expect will define those cell types. So this took us like a year to do it for placenta. And you'd think, oh, well, they have, they're in this space, there's something called ontologies. So it's where experts have done this already to say, here are the main cell types, and here's how they relate to each other. But for placenta, this was totally incorrect, which I thought was crazy that like someone had put it together, I think based on mouse, and they're like, oh, it must be the same in humans. But it turns out, and I didn't know much about placenta. Actually, I know like nothing about placenta before I became a professor, because I was, doing cancer and other stuff. So I'm still learning this stuff too. But that, that placental biologist experts that I collaborate with were like, this is totally wrong. We cannot use this. This is the mouse placenta is totally different than the human placenta. So they can't even really do, some people do, but generally the mouse, like as a model, is not a good one for placental biology, because the placenta has a different structure and has different things. So this had to be completely redone for the placenta. But again, I just wanted to emphasize that it's important, because if you were using a mouse one and then now you're trying to map your human stuff to it and it was totally wrong, it would just be junk, and you would have wasted all of your hard times or hard labor doing that. So anyway, there's a lot of important work that happens behind the computation that's important to acknowledge. Okay, and so for our placental project, we're generating multiple different types of data. So we're the Department of OBGYN, we have patients. So we're actually recruiting pregnant women and early in pregnancy, so that we have all of their clinical data. We have, for some of them, a subset, we actually have ultrasound data and some MRI data of the placenta during pregnancy to link to this, which is pretty cool. And then also doing just a standard histopathology of the cells, which is what they do. If there's a problem, that's how they diagnose it, right? So we have like baseline, this is what they do clinically. There's the MRI and ultrasound. So we have in vivo imaging. And then we are doing, take the chunk and send the whole thing to look to see what RNA, what genes are there, what the chromatin looks like. We even have extracellular matrix, where we're looking between the cells to see what's there and how that's different in different regions. and then looking at the different proteins. And then also we're doing gene expression and the chromatin conformation at the single cell and the single nucleus resolution. So one of the important points of the placenta is that one of these cell types that layers that villus structure that helps with the exchange is multinucleated. So it's a bunch of cells that then fuse. So there's no cell membranes, but there's a bunch of nuclei in this like layer. And so if you took single cells and tried to separate them out, you couldn't see these. And so we take just the nuclei and profile those. And then we're doing the spatial proteomics and spatial transcriptomics, and then looking at the gene expression at spatial scale relative to different structures. So here's an example of one of the MRI images that they've done for the placenta. So these are with our imaging collaborators. To me, this just looks like, I don't know, this doesn't look like anything, but there are, experts that have large computational skill sets that analyze these MRI images to even make the images and then to analyze them. And the goal here is what we've done is then we've used these MRI images in vivo, so while it's alive, to model the placenta in 3 dimensions. And what we see here is like the main part here. This is where it's attached to the uterus. And then there's this membrane that's on the fetal side, that kind of protects it and holds it in. This is what the basis was for this 3D model. That then we'll say, okay, well, we took a chunk from this little area here, and that's where, we've put it into this coordinate framework. And so, but also part of this, right, is when we see the placenta, right, in vivo, it comes out looking totally different. So part of this is difficult because of the sampling, right? It's like when they're actually sampling during delivery. So people are in the delivery room, whether it's a cesarean section in the operating room or if it's a regular delivery. And then having to take it and say, okay, well, where was it? How was it attached? And then they put little pins in with little markers to say, okay, this was up, this was down, right? Because the placenta can be pretty much anywhere in the uterus, and it changes for every pregnancy. And so trying to record that information for us to then go and geolocate it, which I think is pretty cool. Okay, so I'm going to take a couple minutes here for you guys. We're doing pretty good on time for you guys to have some interactive time to go check this, to go look at this, because I think it might be something fun that you could possibly use in some projects you had someday, or you'd remember, oh yeah, there's this great resource. So I'm going to put this in the chat. Oh, you already have it. Hey, look, that's amazing.
Gotcha.
So if you're great. So if you can go ahead and go to that website, and you'll have to scroll down just a bit to the data viewer. You can also link it here. It's really annoying though, it's small, and I couldn't figure out how to make it big. So, but you can take the drop down and you can check the different organs, spin it around. And then on the next step, so that may not be as thrilling as I was hoping it would be. So then you can also go to the functional tissue unit explorer. And this, I don't think is super fully developed yet, because I know what they want to do with this and it's not what it's doing right now. But I like it because you can see on the left side here, within each of the organs, they have what they call functional tissue units. So these are, again, defined by experts that, okay, here are the main structures in the organ that have some distinct function. And so you can click on them, you can see what they look like here, which I think is kind of I can't tell if it's a cartoon or if it's actually like a cell slice that then they've somehow digitized. You can see what it looks like, what the cells are. And then if you click on this side, you can see, what kind of cells are there, what biomarkers are on and off. So you can imagine if someone said, hey, go look in the kidney, in the nephron, and tell me what genes are highly expressed in that tissue. Like, you actually now, because you have this website, can do that, which is cool because you don't need to have any computational skills to do it. And the upstream of this took like years. So yeah, so I'm going to stop talking for like, let's say 4 minutes right now. So you guys can go. And I would like for you, if you can either just put it in the chat when you're done, answers to your question. So what organ did you choose to look at and why? And then how many anatomical structures did your unit have? So that would be from here. And I guess that's the same question, kind of. I was hoping it would be here, but yes. Okay. So how many different things is your organ have? So yeah, go take a couple minutes to play around. All right. How did you do? Were you able to find anything?
Yeah, I did. I looked into the spleen, um, and I just found right now that the female, same as the male spleen, they have six anatomical structures, and for FTUs, the website isn't really like opening, but yeah, I looked into the spleen.
Very cool. Yeah, that's great. Yeah, and that's the other thing I forgot there, the fact that they split it into male, female, and then the data itself, I don't think they necessarily give you all those options on here, but have all the different clinical data associated with it too. So some of them will be like, this person was a smoker, or this person, they're not as normal as I was hoping they'd be actually. But there's, so you can actually see other things. Well, I mean, there's not a lot of samples though. So for each one, right, there's probably a handful of samples. So there's not a lot to do, like what happens when you smoke to your spleen. But you can see additional data types too, which are interesting.
Awesome.
Okay.
Allison, do you want to share too? No pressure, but just want to give you the floor.
Thank you. I checked out the left knee and the right knee for both female and male. The left knee for male and female both have 16 uh, anatomical structures, if that's how you say it. I picked this one because I went on a run this morning and my left knee started bugging me. So I was like, oh, let me check the structure of my knee. But I think it was pretty cool. It was a bit blurry, but it was nice to see like how it is and how it's structured.
That's awesome. Yeah. So I think having this, I mean, it will keep getting better, right? These are still like in development, but I think once the program officially ends in like a year and a half. So, there's like this final push of like, hey, we got to get these websites fully working with all the data behind it. So that's very cool. Great job, both of you. All right. So moving on. So now I'm going to briefly talk about, I'm not going to go in super depth, but I just wanted to introduce you to some of the molecular data types and what they look like when they've been analyzed that we have. So one of the technologies is bulk RNA sequencing. So this is looking at gene expression. And so this is when we're taking, in this case, just a chunk of the tissue. We're not looking at the single cells. And, in some cases, you're taking two different, this is a really gross picture. I just realized. That's like grossed me out before, but I just decided that this is a gross picture. You take, in some cases, you can take two different conditions, so like cancer and normal, take the chunks of tissue, isolate the RNA from the individual cells, but it's going to be a mix of the different cell types. And then you can read the individual sequences and then determine what genes they came from, count them, and then determine if the gene is high or low in the different conditions. So for the placenta, we've done this and we have, so these are MDS plots, so multidimensional scaling plots, where we're looking at the variation in different clinical things. So in this case, we looked at fetal sex, which has a huge difference on gene expression, which is to me was surprising because in humans, depending on the organ and the tissue, that's not necessarily the case, but in placenta, it's a huge difference like from the gene expression level in what boy babies are doing relative to what girl babies are doing, at least their placentas are doing. And you can see that pattern here. So when we looked at across many of the thousands of many of the variable genes, so ones that are changing a lot, we can see very clear distinction between the female babies and the male babies just based upon their gene expression in the placenta, which is something then for all of our other analyses, we have to make sure to account for so that we're not just seeing sex differences and that we're actually seeing other differences. We can also see a very distinct difference in mode of delivery. So those that had a cesarean section versus those that had vaginal delivery. And this is it's expected, but it's also really cool because it shows the biology of something is happening, right? When the placenta undergoes labor, right? So it's being squished by the uterus during contractions. And it takes time. It's not just 1 minute it's in and 1 minute it's out. It takes a long time. So there are changes that are happening in the placenta itself that we can actually see just based on the gene expression. I mean, this can be too, some of the genes changing before labor that actually initiate labor, right? So there's still, which is also shocking. It's not super clear what starts labor. which, you know, you'd think in 2025 we would know this, but we don't. So that's still an active area of study of what's changing to say the baby's like, hey, I'm ready to come out, start having contractions, get me out of here. You know, so this is kind of the first step to starting to understand that. And then the cesarean ones were unlabored, so they did not undergo labor. They were, you know, delivered by cesarean before they had labor. But then what was more surprising to me, I guess, was that we had 3 different regions. So the side closest to the baby, the middle of the placenta, and the side closest to the mom. And those didn't have very distinct gene expression differences, where I would think, well, the ones next to mom are doing something different than the ones far over there, but it's not as big of an effect as we expected. So, you know, you find exciting things all the time. So when we look at the chromatin accessibility, this is where we look to see where some of the chromatin is like wound up and bound, or if it's open. And if it's open, it allows for genes to be expressed. So when we look at these same things, but just at the chromatin accessibility, we also see a very large difference in the sex of the baby. We don't see as much difference with delivery, but we see, so these clouds here, these are tightly clustered. These are actually from the same placenta. So these are the different regions of the same placenta that, you know, we have in here. So you can see there's a very distinct individual signal, like the person, or, you know, the placenta it came from is much more, like, it's not as different than it is across the different groups. And again, the location didn't really have a pattern. The biggest difference was fetal sex. I mentioned we also profiled what's in between the cells. And here, this is what we call a heat map. So we're looking at the individual values, so the levels of these proteins. So these are looking at different proteins in different parts of the ECM and the extracellular matrix. And red means there's a lot of them, so high expression of these proteins. Blue means low. And what is interesting here is that, so the cellular, so we have, this is like within the cell itself. This is what the protein is doing. And then in the different components of the extracellular matrix, whatever these proteins are, you know, are not as highly expressed, but then these ones that aren't part of the cell itself are very highly expressed. So there's all sorts of interesting stuff that can be done with this. And so then when we go into the single nucleus technologies, sorry that PowerPoint made this fuzzy. Now we're taking, you know, this chunk of tissue and dividing it into single cells. So dissociating it, mixing it up, doing whatever to make the cells float around. Then we, you know, sort the cells to make sure we pull out any dead cells and just keep the ones that are alive. And here, these are the target cells. Then we individually sequence the cells and all the RNA within the cells. And in this case, we did the RNA and the attack, like the chromatin accessibility for every single cell. And when we do this, we see, so this is a picture, just kind of a cross section of these cell types right at that maternal fetal interface there we were talking about earlier. We can see each one of these dots is the expression, the gene expression of a single cell. And based on what genes are expressed, we can identify what cell types they are. So this big cloud here are those multinucleated syncytiotrophoblasts. This is that word, it's a big word that I can never spell. That's the one. So we have all these nuclei that we pulled out, and you know, because that's a lot of what it is. So all these nuclei we captured were from this layer that does this nutrient exchange. But we also see some of the endothelial cells, so some of the actual vessels, which are of interest. EBT, so these are the ones that actually invade the spiral arteries. We didn't capture many of those, and what we've decided is that it's because it's a technical issue. So it's not that they're not there because we see them in the spatial data and we know they're there, but that because of the way that they're bound into the extracellular matrix, they're kind of held on by like this fibrotic tissue, that when we did the nuclei extraction, we didn't loosen them enough. And so they just didn't get to the downstream part. And that's a really important consideration for any of the downstream computational stuff, because if you just assume, well, look, there's this little tiny bit of EBTs, that means they're not there. That would be an incorrect assumption. It was actually a physical technical issue that we encountered as to why that didn't happen. So really understanding what happens upstream for any of these computational things that we do are super, super important in understanding the biology, because you know, you can go off on a tangent, you know, go off very in the wrong direction if you don't understand the underlying biology. And so we did see some differences by location at the single cell level or single nucleus level. So just for example, this little tiny cluster here, these are the endothelial cells. And so in the maternal side, we see more of the lymphatic endothelial cells, so ones that are associated with lymph nodes, which makes sense because there are many of those in the uterine, in the endometrium. But then in the fetal side, we see vascular endothelial cells as the highest cell type, which makes sense because that's what they should be doing. And so these would be the ones that are derived from the fetus. These would be the ones that are derived from the mom. So we actually have two different cell types from two different individuals, like in this one analysis, which is cool. So we can then, do further analysis to see, okay, well, how are the cell types interacting with each other if they're from two different people, basically. And so then we have the spatial transcriptomics. I'm not going to go into the details of how this works, but what we're seeing here is a cross-section, like a slice of the tissue. And then each of these spots, this is the Visium technology, so it's kind of overlays a grid onto your tissue. And then each of the spots, you can read out the gene expression from each of the spots and then determine what cell type is which. You can see in this technology, though, the placenta is kind of spongy, so there's a lot of space. So all this white, or not white, beige space in here is just empty. That's where that would be the intervillous space. That would be where the blood would pool to from the maternal blood would pool. And so this technology didn't work great for us because we're like, well, there's a lot of blank space. But when you zoom in, here's an example of an endothelial cell, so like a blood vessel, and defined by this gene. And when you zoom in, you can see, okay, right in the middle of it, we've got really high expression of this one marker. So this is most likely an endothelial cell. And then you can look to see what's near it. A higher resolution technology we've used is the Genomics Digital Spatial Profiler. Again, I'm not going to go into deep detail about how you do it, but again, you take a slide and you have fluorescent antibodies that you then image, and then you have this beautiful picture. So here we took different parts, like I told you, different parts of the placenta, made these nice little slides. And then we make these beautiful images, a full cross-section of the slide that we have. So in this case, this is the basal plate. So this is the side connected to mom. And this would be, going away from mom. And what we can see here is we define individual spots that we want to look at. So we don't, because it's big, we don't get to see what's in everything, at least with this technology. So we have to choose what we're looking at. And so we choose these different regions to profile. But then when we do, we get this beautiful image of, so in this case, these green things are the extravillous trophoblasts. So these are the ones that are invading the maternal spiral arteries. I think I can play this. Can't I play this? And this technology then can actually profile the gene expression from just the cells themselves. So see that red part, that mask is saying, don't look at gene expression there, just pull the things out of these green cells. And so then when we get the gene expression, we can say, okay, this is what the extravillous trophoblasts are doing specifically based on this and where they are. So it's a pretty cool technology to be able to do, but then you can imagine all the data that you have. Here's a view of the villi. So here we have the syncytiotrophoblasts surrounding the villi. And here the yellow are the blood cells. You can actually see the individual red blood cells in here. And these red things are the actual vessels. So you're looking at a cross section through one of these cauliflower stalks with the blood in the middle and the trophoblast around the outside. So you can imagine with this technology and computationally where you can say, okay, well, what's the distance between this villi and this villi? And does that change how things are being expressed? And here's an example of a larger vessel, an endothelial cell vessel that we are extracting the gene expression from. So anyway, I think these are beautiful. And so some of the things we can do, right, look at different cell types. And again, this is based upon what we know, right? So it's only as good as our markers are good. And then from the protein level, we also use imaging mass cytometry. And this is a different technology, but produces, at least to me, similar looking images, but they're not, where you have, different stains for different cell types. Again, you can see like here's another cross-sectional one where you can see in the middle here, this is likely the vessels and the different cell types. And so you can use various imaging analysis techniques and computational to define the cells, to look to see how cells are communicating, look at the various levels between cell types. So there's just an example of what the fetal surface looks like, the maternal surface looks like, kind of different by eye. Pretty pictures. Okay, that's just more pretty pictures. I like pretty pictures. But then what you do is once you have these data extracted, you can put it into this similar, analysis pipeline where you say, okay, how do the different cell types cluster out based on their expression of their genes? How do they cluster out based on where they are? And so example, like here we have microvasculature, so little tiny vessels versus like the big blood vessels in the green and the orange, and we can see that they actually express different genes. So it's not different genes, but they are doing slightly different things even at the gene level, which is pretty cool. And here's just a picture. Getting up on time here, so I'm going to go fast through this, but they are actually doing different things. Here, we're not going to go through this right now, but here's another link that is also a really great link to go to look at the molecular data. So before you just had the tissue units and the structures and the 3D models, this link here gives you, these images depending on what you're looking at technology wise, and you can turn on and off some of these various genes, which are marker genes for things, to look at the images. And so one of the drop downs is called DAPI, D-A-P-I. That is a nuclear stain. So when you turn that one on, you'll actually see the individual dots, and each one of those is a cell. And so it kind of helps you to give you more reference than this picture here. There's just this one protein stain. I encourage you to look through it. It's very exciting. I think it's great. And again, you can download data and process it and do whatever you need to it. So it's very, very powerful. And so, yeah, so with that, that's what we're working on to create a 3D molecular atlas of the human placenta at various spatial scales with lots of different multi-omics data types that takes very large amounts of compute power and technical computational skill, but also biological, insight and knowledge and lots and lots and lots of collaboration. So with that, thank you for your attention. I'd like to acknowledge the funders of this study and all of our collaborators and the tissue donors, especially, for being part of this study. And so with that, does anyone have any questions?
Any questions? Feel free. I guess I'll start. I have a question. So in the beginning of your talk, you went through this like journey from what you were interested in high school to like what you ended up kind of doing now. So as you were going through these stages, like what were some of the questions you were asking yourself to figure out what you actually want to do with your life?
That is a great question. Yeah. So I think I, at least in high school, was kind of just on like, being a doctor sounds like a good job. That sounds great. I like biology. I'll be a doctor. So I didn't really have much of a beyond that, right? But at least I had that. So that was good. So I went in as a biology major to Berkeley, which was helpful, at least kind of having something. And biology is diverse, so you can do lots of different. Same with computer science. Same with a lot of these things. If you get a good base, you can apply them broadly. But then, yeah, I mean, I really was pre-med, but then I had the opportunity to have this undergrad research experience. And so that's something I definitely recommend, even in high school, if you have the opportunity to do some research experiences, because the research is not for everybody. Also, that's the other thing, is that like some people love it. Some people are like, nope, I'm going to go be a doctor. This is not for me. And so, yeah, but I think it was really just like, wow, this is so cool. I'm so excited. I have to do this now. For that. But I mean, along the way, there were a lot of hard points. I mean, grad school was awful. Grad school is really hard. I almost dropped out after my first year, but I didn't, because I had really supportive mentors, and I was very, and I told them what was happening and how it was hard, and they supported me. So I think also, making sure that you reach out when you're having struggles and like trying to work through them, not just giving up, I think is one of the things that I've had to do a few times. But yeah, I mean, to me, it was really just following what I was excited about. And at every step, like, oh, well, humans are cool too. Hey.
Oh, that's awesome. Thank you so much. Thank you. Yeah. Oh yeah, Allison, go for it.
I wanted to ask, like, you got into UC Berkeley, and that's amazing. I am also interested in like pre-med, and I would like to know like what steps or what did you do during high school to get there? And when you got to UC Berkeley, you went into that research program. What are like some tips to like get into that, or to be able to apply, or where did you find those programs? Because thankfully I found this program and I really enjoy it. So I was wondering where you find it, what steps did you take, what advice do you have?
Yeah, great question. Yeah, so in high school, I mean, so I got good grades. I took AP classes, but I don't think that's a requirement. I think getting good grades and having extracurriculars. But again, I didn't do any like the undergrads now, or these high school students even coming in and with these extensive research resumes. I'm like, wow, I didn't know that existed. Or maybe it didn't exist. I don't know, not to me. But being active, so having extracurriculars, working toward doing these programs is huge. Getting good grades, having a good essay. I think maybe working early with your teachers to make sure your essay is strong and your package is strong. And then also not worrying about the school, right? Like going and doing it is more important than where you're actually going, right? So Berkeley back then was not as hard to get into as it is now. All these UCs are great, Cal States are great, community college is great, right? But wherever you end up, as long as you're working towards your goal, it's okay. But then I would say in terms of as an undergrad, well, and actually in anything, if you're interested in something, just email people. Cold email people is like the best way to get someone's attention. And you might not get a response from everybody, but a lot, like that's my undergrad research experience was one of my TAs. So like for one of this ecology classes, he was like, hey, anybody in this class, I have this research project. So they want to help me. I need an assistant. But for most of the colleges, they'll have like a centralized database, like UCSD, for example. They have here are internship opportunities where, say, if I need someone, I will post a job posting and say, hey, this is what it is. If you're interested, apply. So just being on the lookout for that. But then also if you're specifically interested in, say, like OB work, you go and look at faculty on websites, at the college that you're at, that do what you want to do, and then email them. Say, hey, this is who I am. I'm interested in this. Do you have any openings or have referrals for me? That would be my recommendation. So always kind of keeping your eye out, also making sure you plan in advance, right? So if you're like, I want a summer position, usually April-ish, start looking, so that it's, if you're like in June, you're like, hey, can I start your lab next week? No, right? Like, so planning ahead and just kind of having a general landscape. But there's a lot of opportunities, and I mean, at least in my experience, everybody's been very, very open to training people and bringing people into research. So I don't think you would have a problem with that.
Awesome.
Thank you.
Yeah.
Perfect. And also, Allison, so heads up, that sneak preview for next week's action items, but part of your action items for the program are going to be to like look into different majors at the UCs and like look into different financial aid options. So like also throughout the program, feel free to ask me any of these questions, and I can either point you to smart people like Katie who are much smarter than me, or I can try to send you some links to resources and stuff. So just keep doing what you're doing and you're on the right track.
Thank you so much.
Yeah.
Yeah, I was gonna say you guys are way ahead of the curve in this program already, I'd say. So keep up the good work.
Awesome. Any other questions before we call it? Feel free to type in the chat. Feel free to raise your hand. Feel free to unmute. No pressure. Awesome. Well, thank you so much for joining us. That was a great talk. I know everyone learned a bunch of stuff, and yeah, I really appreciate the time that you took to present to us.
Absolutely. Happy to do it. It's nice meeting you all. Hope you have a good rest of your day. Oh, and it's Friday. Yay.
Yeah, Friday. Woo.
Have a nice weekend.
Bye, everybody.
All
Have
right,
a
take
great
care.
weekend.
Bye.